And we'll move on to the last speaker of the session, Diana Rennison from the Division of Biological Sciences at the University of California, San Diego. And the title of her talk is Uncovering the Genetic and Ecological Underpinnings of Parallel Adaptation. Take it away.
Thanks so much. And thanks so much to AmNat for bestowing this great honor upon me, especially knowing the long list of leaders of the field that have previously received the award and watching the really amazing work of my co-awardees. So my work over the past decade and now the work of my lab is really focused on understanding the core mechanisms that underlie the really great diversity of species that we see on Earth today. And the work really falls under two sets of questions. The first is, how do sources of selection interact to shape evolution and the generation of biodiversity? And the second, which I'm going to talk about today, is why do organisms follow certain evolutionary trajectories? So this concept of evolutionary predictability is something I'm really interested in. And there's been a long-standing fascination in determining how predictable evolution is. In Stephen Jay Gould's book, A Wonderful Life, he posed this really fascinating thought experiment where he asked, if the tape of life was replayed, would we see the same organisms evolve that we currently find on Earth? And a classic answer to this question has been no, but there's a lot of emerging data in the last few decades that have challenged this view. And we're seeing that there is repeatability from the level of ecotypes all the way down to genotypes, but that it's highly variable. And we don't have a really good framework for understanding this variation in whether or not we see repeatable patterns of evolution or unique patterns of evolution. And interest in predictability also has an applied aspect to it. So knowledge of how organisms will evolve in response to selective challenges like antibiotics or climate change, or in artificial settings where there's agricultural breeding, are becoming increasingly valuable. So I use the Threespine stickleback as a model of parallel evolution. So stickleback are pretty cool because there's an extant marine ancestor which exists throughout the world. And these marine individuals have colonized freshwater environments repeatedly particularly after the last glacial maxima, as new freshwater environments were forming, so lakes and streams. And once these marines encountered these new freshwater environments, they've diversified their niches. So we see different populations that have diversified into limnetic ecotypes that exploit kind of the open water zone of the lake, benthic individuals that exploit the nearshore environment. We have individuals that inhabit solitary lakes, and then also individuals that inhabit streams. And we've seen these limnetic and benthic individuals evolve a handful of times in British Columbia, Canada, and we have these solitary and lake and stream forms that have evolved thousands and thousands of times across the globe. So this replication gives us a really powerful system in which to ask what predicts patterns of parallel evolution. And we also have different gene flow contexts in which this divergence has occurred. So this limnetic and benthic divergence has occurred in sympatry. We also find variation in solitary lakes where some populations are quite benthic, some are very limnetic, and some have phenotypes that are in between. We also have a lot of geographic variation in this radiation of phenotypic variation. So all of the red zones on this map are where stickleback populations are known to occur, and the yellow zones are places that stickleback could occur based on climatic variables. But what you can see is they're widely distributed throughout North America, as well as Europe and Asia. So this allows us to compare the repeatability across different geographic distances as well. So the specific aims of my work are kind of twofold. First, to quantify the repeatability of genetic evolution, and then secondarily to test which ecological, geographical, and genetic factors explain these patterns of repeatability. So I'm going to briefly introduce you to the approach we use to quantify genomic parallelism. So basically, we have multiple replicates of any particular ecotype we're interested in. So in this example, we have benthic and limnetic ecotypes from several different lakes. We then use either whole genome resequencing data, genotyping by sequencing, or RAD-seq data, which are just reduced representation technologies for multiple individuals per lake. And then we estimate FST, which is a genetic measure of divergence, for thousands of variable sites across the genome. And then we quantify genetic parallelism using either a genome-wide approach, which I'll introduce you to, or for thousands of individual genomic regions. So for the genome-wide approach, what we do is we pick a pair of populations. So for example, benthic individuals and limnetic individuals from Priest Lake, and we estimate genetic divergence using FST across the genome. So we have all the different chromosomes. And we can see that there's some regions of the genome that are highly divergent, so large differences in allele frequency. And there's some regions of the genome where benthics and limnetics don't have different alleles. And we repeat this for multiple different population comparisons. And just looking at this, I hope you can appreciate that the patterns of genomic divergence are really similar for independently derived benthic and limnetic stickleback pairs. But looking at these graphs just isn't enough to say, yeah, these look pretty similar. We want actual numbers to be able to quantify the level of similarity between these independently derived pairs. So what we do is we use pairwise correlations where we ask for a single region of the genome, is it the case that one population has a high value of FST and the other population has a high value, or a low value, or a high and low value? And all of those individual estimates give us a correlation coefficient which summarizes how similar the pattern of divergence is for independently derived pairs. For our locus-specific approach, if you pretend that this black rectangle is the genome, we have regions of the genome that are variable, as indicated by a gray snip here, single nucleotide polymorphism. And then we have regions that are invariable, which are indicated in black. So this would be for a single comparison. So for example, between benthic and limnetic individuals in a single lake. So we can classify what's variable and what's unvariable in the genome. Then among the variable sites of the genome, we can distinguish whether or not there's a signature of adaptive evolution. So what we generally call an outlier SNP, which is a highly differentiated region of the genome, which is likely to be under natural selection. And then our goal is to do this multiple times and ask whether or not there's repeatability at a given SNP or region of the genome. So basically, we're looking for regions of the genome where multiple populations where we have data share an outlier, and we designate these to be parallel loci. And then we have other regions of the genome that are non-parallel loci, and then of course we have the invariant regions of the genome. So once we've either quantified parallelism genome-wide or at particular regions, the next goal is to look at different variables and ask whether or not they explain these patterns. So in terms of ecological factors, there's a handful of things that we've looked at previously. So the first is the similarity of natural selection. So this is a pretty gross estimate of natural selection. We've generally looked at different habitat contrasts. So asking if population pairs come from the same habitat type, so both streams, are they more similar in their pattern of divergence than if you compare a stream population to a lake population? For geographic proximity, we've looked at the distance between populations and asked whether or not parallelism correlates to how close together different populations are. And lastly, we've looked at whether or not the divergence between pairs of populations is occurring in allopatry or in sympatry. So whether or not the potential for gene flow affects whether or not we observe parallel evolution in the genome. So we have several predictions, one for each of these different units. So for selection, we predict that population pairs with more similar ecological niches will have more genomic parallelism. And that prediction comes from theory. And for large-scale geographic distance, we would predict that nearby population pairs will have more genomic parallelism because of more similar founding populations and more similar abiotic factors. Unfortunately, because we're surveying wild populations, we can't disentangle those two different hypotheses. And for geographic context, we predict that sympatric population pairs will have more similar genomic parallelism because of genomic constraints that are imposed by high levels of ongoing gene flow. And what I mean by that is that some of the previous work that has been done in stickleback has shown that the probability that you'll see a locus contributing to adaptation is dependent on the recombination rate of a particular region of the genome. And that pattern differs whether or not the populations are undergoing divergence with or without gene flow. So without gene flow, we see that the probability of an outlier contributing to adaptation, or a SNP contributing to adaptation and being an outlier, doesn't really depend on recombination rate. But when there is gene flow occurring, it's really, really heavily dependent on the recombination rate. And you really only see outliers where there's really, really low recombination, which means that a smaller fraction of the genome is available for adaptation to occur. And thus that might bias the pattern that you see and lead to a higher degree of repeatability. So to look at the effect of ecology and geographic distance, we basically set up a whole bunch of different contrasts. We chose limnetic populations, benthic populations, solitary lake populations, and stream populations, and had multiple replicates where we compared them against a common marine population. So the key here was that we were always estimating divergence of these different freshwater ecotypes against a common marine comparison. And then we were able to ask whether or not the parallelism differed depending on a bunch of different variables. So we used this correlational approach. So in essence, we had all these pairwise correlations. So for example, we would have one marine limnetic comparison and then look against another marine limnetic comparison and estimate what the correlation was. And then we would also do this so in this case, we had the freshwater populations having the same ecotype, same ecology. And then we also had comparisons in which the comparisons would have different ecotypes and different ecology. So we were able to kind of tease apart the effect of the similarity of the niche to how strongly the genomic patterns were similar. The populations that we looked at also were collected from around the world. So we had differences in geographic proximity for our different samples. So we had some that spanned along the west coast of North America, and then we also had populations from Europe. So we were able to use continuous estimates of distance and also look at the effect of ocean basin on patterns of genomic parallelism. Okay, so jumping into the results. So first looking at the effect of ecological similarity. So on the y-axis here, we have the genome-wide FST correlation coefficient, which was estimated how I explained to you previously. And we'll start by looking at populations that were found in the same ocean basin. And what we found was that the correlation coefficients were significantly higher for population comparisons where the freshwater individuals were from the same ecotype compared to a different ecotype. So this suggests that ecological similarity, what we think represents similarity of the selective landscape, is predictive of how similar patterns of genomic divergence are. When we looked at populations that were collected in different ocean basins, we found that if you look within the same ecotype classification, the levels differed significantly. So they were much higher when population comparisons were from the same ocean basin. So this is suggesting that populations that were nearby, either due to similarity of abiotic factors or of the founding marine populations, had much more similar patterns of genomic divergence. And again, if we look within different ocean basins, we still see this effect of ecological similarity. So this suggests that both ecological context and geographic context are really important for predicting the level of genomic parallelism that is observed. So recently, a postdoc in my lab undertook a study where we looked at the effect of gene flow. So as I mentioned earlier, you can find limnetic-like populations evolving in allopatry and also in sympatry with benthic ecotypes. And so we looked at 3 replicate pairs where there were independent evolution of limnetic and benthics in either allopatry or sympatry. And then we asked whether or not that affected the level of parallelism that we observed. So again, we use this correlational approach. So here we have a pair of benthics limnetics that were allopatric and another allopatric benthic limnetic pair. And as you can see, there's essentially no correlation in the pattern of FST across the genome. Whereas if we look at 2 different benthic limnetic sympatric species pairs, we find a really strong correlation. And so we've repeated these analyses and we were able to summarize the overall patterns. And we found that the pattern of genomic parallelism was much, much stronger for the sympatric benthic limnetic species pairs compared to the allopatric pairs, where essentially they were each doing their own unique thing in terms of generating those phenotypes using different genotypes. And indeed, when we compared the pattern of divergence for allopatric pairs to sympatric pairs, we found, again, no correlation, which suggests a distinct genetic basis for producing the benthic and limnetic phenotypes. Okay, so that's kind of the genome-wide pattern and the different ecological factors that we considered. But I'm also really interested in genetic factors and how that can affect patterns of reuse. And there's a handful of different factors that I'm interested in, including pleiotropy, mutation rate, recombination rate, and the source of genetic variation. And so people in my lab are working on these different factors. But today I'm just going to tell you about pleiotropy, which is kind of one of my favorite aspects. So pleiotropy is when a single gene influences multiple traits, independent traits. And pleiotropy has been something that's been suggested to be a source of constraint during adaptation. And that fact that it could contribute to constraint means that it could contribute to patterns of repeatability or gene reuse over the course of adaptation. And in 1930, Fisher produced a model which suggested that the probability a mutation would be favorable was dependent on pleiotropy, which he termed mutational size. So he suggested that if a locus was more pleiotropic, it would have a lower probability of being favorable. And the reason for this is just that if you have a single gene influencing multiple traits, the probability that it will be favorable on multiple individual traits increases or decreases really quickly because it's difficult to actually have that be positive on the overall adaptive landscape when you have an increased number of traits. So kind of extending this idea, we would predict that loci with reduced, so lower levels of pleiotropy, would be more parallel because they'd be more easily used repeatedly in each independent population. Now, pleiotropy is really difficult to estimate. If you wanted to do it perfectly, you would pretty much have to knock out each gene in the genome independently and then measure every single trait that it could ever affect, even things you couldn't necessarily think of. So we have to use proxies to estimate pleiotropy. And I used 2 different proxies. So the first was gene connectivity. So work in yeast and other laboratory model systems have suggested that gene connectivity actually predicts real pleiotropy, so how many traits a single gene affects. And so I built gene co-expression networks to estimate gene connectivity. And then I'm also super fortunate because people have been working on stickleback for a great period of time and are really open with data sharing, that there's been numerous QTL, so traits mapped to individual regions of the genome, and that these have been compiled into a database that I was able to use. So I was able to look at hundreds and hundreds of QTL and ask how many traits map to the same region of the genome as another estimate of pleiotropy. So these are my actual estimates of pleiotropy, my proxies. using QTL number and gene connectivity. And so the first thing I just want you to see here is that there's variation in pleiotropy in the genome. So many of the genes or genomic regions have really low levels of pleiotropy, so they're not very connected, or they only influence a single trait. However, there are regions of the genome that are super highly connected or influence several different independent traits. So this variation means that it's possible to test whether or not it predicts patterns of parallelism. So to classify parallelism, I used our independent benthic limnetic replicates and also streamlake replicates. And I had 3 benthic limnetics and 16 streamlake pairs. And I had tens of thousands of SNPs for each of these pairs. And basically using that individual locus method that I outlined earlier, I was able to label these regions as either parallel or non-parallel. So we have these 2 different types of loci in the genome. And then we asked whether or not the amount of pleiotropy differed between them. So starting with the benthic limnetic data, we have QTL trait number on the y-axis, and then we have parallel loci and non-parallel loci. And what I hope you can see is that the parallel loci actually generally influence a greater number of traits than the non-parallel loci. So this is opposite of Fisher's prediction. It wasn't that trait number was minimized to increase repeatability. And we found the same pattern for the streamlake comparison. So on average, the parallel regions of the genome influenced 2 or 3 traits, whereas the non-parallel regions influenced 1 or 2 traits. And when we used connectivity as our estimate, we found the same pattern. So on average, in parallel regions of the genome, there was more highly connected genes than the non-parallel regions. It wasn't statistically significant for the benthics and limnetics, but it was for the streamlakes, but the pattern held in both cases. And this isn't an effect of gene density, so there's no significant difference in the number of genes in these genomic regions. Okay, so to kind of summarize the blitz of results that I've given you, first is that we find considerable parallelism across the genomes of independent stickleback ecotype pairs. So that's exciting because that means that we're able to look at the mechanisms that influence variation in these patterns. We find that parallelism is highest when ecological conditions are most similar, and that matches a theory that suggests that as the selective landscape becomes less parallel, you should see decreasing genetic and phenotypic parallelism. We find that geographically close populations exhibit more parallelism. Parallelism's much higher among sympatric pairs than allopatric pairs. And the parallel regions are more pleiotropic, which is counter to theory. So this means things are repeatable, yay. Maybe we can actually predict evolution. I already mentioned the other thing. The effect of geographic proximity, we think, is likely due to patterns of standing genetic variation in the marine populations. So in stickleback, the use of standing genetic variation during adaptation seems really, really key for rapid evolution. And so we think that populations that are geographically close to one another probably had more similar marine populations found them and more similar initial pools of standing genetic variation. And since those loci were being used for this rapid adaptation, it kind of constrained or influenced the pattern of gene use and led to higher patterns of parallelism. We find the effect of gene flow. We think it's probably due to this recombination rate bias that we find when gene flow is high that leads to higher parallelism. And our results suggest that low levels of pleiotropy might not be constraining, which is exciting. And we're following up on this work by looking at whether or not pleiotropy is synergistic or antagonistic. So I didn't do this work alone, and I have many people to thank. So much of the work was done in Katie Peichel's lab while I was a postdoc, and in collaboration with Yoel Stewart and Daniel Bolnick, in which I have ongoing collaborations with, which I'm very grateful for. Andy Har is my postdoc, and he's done a lot of the most recent work. And he's also giving a talk this afternoon on gut microbiome parallelism, so you should check that out. The work was also done in collaboration with Dolph Schluter, Kieran Samek, Greg Owens, Kira Delmore, and Sarah Miller. And I have to offer huge thanks to the broader stickleback community. These are really large data sets that have been generated by many, many different independent labs. And I'm super grateful that the community is really an advocate for data sharing and data archiving, which is really phenomenal. And there were several institutions and agencies that funded the work. And thanks so much for listening to me today.
Thanks for that really nice talk. There are several questions that came in. The first one, this from Moises, does the gene connectivity correlate with how many times the gene shows up in QTLs?
I haven't looked at that. Oh my gosh, that's such a good suggestion. Yeah, I haven't looked at that. I should though.
The next one is, was the co-expression network time and tissue specific?
So there were I used several different data sets to generate the gene connectivity estimates. So I built it off of Streamlink data or benthic limnetic data or just stream data or just lake data. And I used different tissues, and it was robust to what was used to build the connectivity model. So I don't think that matters, but we looked into it.
Great. Another comment, a really cool talk. I am interested in pleiotropy and historical contingency. Is there a way to evaluate bias on the traits people have looked for QTL? I would suspect that some of these traits are linked to the repeated evolution of these ecotypes.
So I'm not sure that I'm understanding the question correctly. I mean, I think that's what we're trying to get at, in that how these mutations occurred. It's not that we necessarily think that it's a single mutation in each of the genes that's influencing these multiple traits. It's that the gene probably has multiple mutations that are influencing multiple traits. And so, I mean, the order of the mutation and those aspects of the historical contingency definitely would influence the patterns that we've seen. We want to dig into the actual individual loci a lot more in the future. And we're planning, depending on if NIH wants to give us money, to do manipulative experiments where we actually look at a single locus and a single mutation at a time, and then look at how many traits and what the fitness effects are of those individual ones, and how that looks, how that predicts patterns that we see in nature. But I think right now, with the data that we have, it's impossible to kind of go down to the finer scale that's necessary to look at the type of question that you're suggesting. But I think it will be possible in the future with both manipulations at the genetic level, so genetic engineering of lines in which we have a common background, genetic background, and then just have differences at individual genes. And then also more high resolution sequencing data.
And this question might be related to that. Could the pleiotropy effect be due to selection on suites of traits in each habitat?
Yeah, so I mean, that's what we want to look at with this synergistic versus antagonistic pleiotropy. We want to ask whether it's the case that we find more pleiotropy than is expected by the theoretical work, because it's the mutation is influencing first dorsal spine, second dorsal spine, and third dorsal spine length. And all of those things would be favorable together versus, you know, something that's increasing dorsal spine length but decreasing eye size, or something in which you might not actually want those two things to increase in the same direction in the same environment. And so I think what's really key here is how the mutations are influencing these individual traits, and whether or not it's a suite of traits being favored or a hodgepodge of things together.
There are a number of other questions, but we're past time. Sue, do you want me to keep asking questions, or?
I think maybe the best thing to do is to ask the speakers to save the chat so they can see the comments and questions that they receive, so that people can have a break before we start the next session in 45 minutes, or roughly that, I guess. Or is it an hour and 45 minutes? Anyway, in the future.
An hour and 45.
But I'd like to, first of all, give a round of applause to Diana for that wonderful talk.